Good morning to all of you. Today I am going to discuss with you about the somaclonal variation. Somaclonal variation, it is given in your plant biotechnology syllabus. Just after your protoplast culture it has been given. Now what is the basis of somaclonal variation? Somaclonal variation occurs as a result of your genetic heterogeneity. Genetic heterogeneity here refers to the change in the chromosome number or the structure of the chromosomes. Now, heterogeneity also occurs during protoplast culture when we are fusing two protoplasts from different species to make a hybrid protoplast, and due to its heterogeneity, one of the DNA content or the chromosome content in protoplast A, they get almostly loss of all of their part of their chromosomes and makes us hybrid. Now this type of things can also observe in your protoplast culture, and during extension of culture periods can also leads to this type of condition. Why this occur? Firstly, it has been saying that expression of chromosome mosaicism or genetic disorder. Okay, this can be your reason of somaclonal variation. So due to expression of genetic disorders, it can leads to somaclonal variation or spontaneous mutation during culture conditions. So spontaneous mutation can arise during extended culture periods. Then also other things are also there, like what type of explant we are using, that can also leads to this type of variation, like meristematic explants. Meristematic explants like your apical meristem, which are derived from the tip of the shoot or axillary bud, they have a less degree of genetic variability comparing to the non-meristematic explants. Okay, non-meristematic explants, non-meristematic explants, they show great degree of genetic variation. Why? Cells of meristematic explants, they divide by normal mitosis and they are maintained at uniformly diploid level, while the cells of non-meristematic explants, they are derivatives of the meristematic explants, and they do not divide by normal mitosis, but they undergo DNA duplication, endo-reduplication, and endo-reduplication result in endo-reduplication means duplication will occur, but cell division will not occur. So duplication of the DNA will occur, DNA content will occur, but that is the S phase will occur with, but the M phase or the mitosis phase will not occur. So due to this, it leads to the formation of chromosomes having 4 chromatids, having 8 chromatids. That means polytene like condition, polytene chromosomes like condition, it will occur. So this type of event can also leads to somaclonal variation. Extended culture periods can also leads to plants regenerated from tissue culture having some chromosomal abnormality. So what it is written here, the genetic changes associated with somaclonal variation includes your polyploidy, means having multiple sets of chromosome, aneuploidy, chromosomal breakage which leads to chromosomal mosaicism, deletion, translocation, and gene amplification, besides several types of mutation. In fact, presence of several chromosomal aberrations means chromosomal structural change like translocation, that is shifting of a segment of a chromosome to different location in another chromosome. Deletion means remove of a segment of a chromosome and gets uh, deleted and gets inactivated due to not having centromere in them. Chromosomal breakage, inversions, multicentric acentric fragments have been found among the somaclones of barley, garlic, and oat. So these type of events have been observed in these plants, barley, garlic, and oat. They have found to be of variations. Now mutation in culture is relatively very low, it occurs very low, and mutations may be due to the what type of nutrients we are using in the tissue culture medium, what type of culture conditions we are having, and mutagenic effects of the metabolic products that are produced by the plants that are getting accumulated within the culture medium, that also leads to your mutation. Okay, now molecular level, if you see from molecular level of view that you have studied in your semester 4, um, sorry, semester 3, your molecular biology part, if you see, you have heard the term of transposable elements, the jumping genes that shift from one place to another place within the same chromosome or from the different chromosome. So due to transposable elements also, then the mitotic crossing over and change in the cytoplasmic genome have also been reported that this type of events can also lead to somaclonal variations. Now let us see the nomenclature of the somaclones. Somaclones that are regenerated from tissue culture, they are regarded as R0 plants, or you can say R plants. So this is the nomenclature of somaclones, that the plants which are regenerated from tissue culture, the somaclones that we have regenerated from the tissue culture, they are called as R0 plants or simply R plants. Then the self fertilization of these R0 plants occur, and after self fertilization the progeny that we get, the baby plant that we will get from that R0 plant, they will be represented as R1 plants, then after that R2 plants, R3 plants, R4 plants throughout the subsequent generations. So in this way we normally classify the name, we do the nomenclature of these somaclones. Some workers use the term as SC, S for somac, C for clones. Instead of using R name, they use this term SC1 in place of R0. That is the first somaclone, that variation we find. Then a progeny SC2, SC3, SC4 for the subsequent generations. Simply, it's not so difficult for nomenclature of somaclones, what type of name we give. Either we give R or we give SC. Okay, just remember the first somaclone that we have derived, that we have regenerated from the tissue culture, will be represented as your R0 or simply R plants. Okay, and then after self fertilization of this R0 or R plants, we get the progeny plants, the baby plants, they will be regarded as R2. Then after the next generation, it will be regarded as R3, then R4, and so on in subsequent generations. We can also use this term of SC instead of using the R. Okay, now the main question arises that how we can isolate the somaclonal variants. Different ways are there, and we have to look through these ways, different ways. So there are two procedures for commonly used for obtaining the crop plants having somaclonal variation. Now why we are doing this type of variation, why we are changing the chromosome number, due to these factors, taking the non-mesomeric explants, it's mainly that maybe due to mutation we may get some beneficial expression of the genes, okay, uh, that are helpful in agriculture aspect. So let us see, we will also do the applications of somaclonal variation later on. So there are two procedures for your somaclonal variation. One is your with in vitro selection and another is without in vitro selection. So two ways are there, with in vitro selection and without in vitro selection. Let us see how we can go for this somaclonal variation without in vitro selection first. Let us see. Image has been given. We try to explain this without in vitro selection first. An explant such as leaf, stem, root is cultured on a suitable medium. Okay, so we are culturing them in suitable medium. Preferably we use MS medium, Murasi's and Skog medium, which is supplemented with growth regulators. Okay, so we take an explant, and that explant is cultured on a suitable medium that we normally do in tissue culture. Then we get unorganized callus, which are the unorganized mass of cell parenchyma in nature, and cells that do not contain any selective agent. So we use such type of callus that we derive from that explant that have no such any selective agent. That means any toxic agent or inhibitory agent should be avoided. Okay, naturally. Now next it is saying that these cultures are normally subcultured. Okay, so these cultures are normally now subcultured and transferred to a shoot induction medium for the regeneration of the plant. So here no selection medium is used. What type of variation you want to them to do, whether you want to them to selectively be making them resistant to certain toxic chemical or inhibitor, that type of selective agents are not given here. That is why it is written without in vitro selection. So we are not using any selective agent in this tissue culture medium in this without in vitro selection. We are making them actually in you will see them in vitro selection. We will see later on that there we will using selective agent and forcing them to induce some chromosomal change so that they can withstand in that condition, in that selective conditions. But here we are not using any selective agents. We are just subculturing them, transferring them into pots. That means we are doing hardening. Okay, and we regenerate the plants. The so called produced plants are grown into pots, transferred into the field, and then they are analyzed for somaclonal variants. Now how they are analyzed? They are analyzed by cytological study. They are analyzed by many ways. By you can go for gel electrophoresis. We can go for gel electrophoresis for studying the variation in their DNA content, the DNA bands that we will get in gel electrophoresis in agarose gel. We will see them and we'll check them for if some variation occur in the plants or not. So here we are not using any selective agents. Okay, we are just we do the tissue culture, similar procedure we are doing the tissue culture here, and we are trying to see later on that if some variation occurs or not within the within that regenerated plant. So here this is the explant we see, and from that explant we do culture in the tissue culture medium, and after certain days we regenerate callus here, unorganized mass of cell, and from the callus we go for shoot induction medium. This is simplified diagram. Shoot regeneration, here no selection is carried out. Okay, without in vitro selection, no selective agents like certain antibiotics or heat stress, okay, or salt or excessive inhibitors we are adding. We are not using that. Then after shoot and rooting we are doing hardening, and then transfer this is the hardening step, and then we will transfer them into the field, and then we try to screen them. We check them for our desirable characters, that whether our desirable characters has been induced or not. The environmental conditions is make in that way, but we are not adding any selective agents to the medium. Just we are just we are changing the environmental conditions. For screening, what we do, we have to analyze their DNA by resolving them in gel electrophoresis, or we can also go for the isoenzyme analysis. Isoenzyme, okay, isoenzyme analysis we can also go. Okay, in isoenzyme analysis we are actually resolving the proteins just like an SDS page. Okay, sodium dodecyl sulfate gel electrophoresis, just like that we also analyze the isoenzyme by doing the certain type of SDS page which is known as native page, where SDS is not used in that electrophoresis. Just we have to separate the proteins based on their structure and size in the gel apparatus, and then for specific isoenzyme analysis we use certain chemicals. Isoenzymes are what? They are certain enzymes which are functionally they are similar, but their structure is different. Now it varies from one plant to another plant. Okay, so as their structure are different but their function are similar, so we can resolve them in gel electrophoresis by a modified way which is known as native page. Native page, in native page, what we do is we resolve the gel, we use the gel apparatus, we make a gel with the help of your agarose. Okay, we use agarose, and we also use certain other chemicals as well, like your um, different type of chemicals are used, and then with the help of the pore size present in that agarose gel, we separate the proteins based on their, here we are separate the proteins based on their size also and on their molecular weight. In SDS page we are using SDS to denature, to break the secondary structure of the proteins to make a linear form. Okay, but here in native page we are retaining that structure, and we go for that isoenzyme analysis for observing that whether certain changes has occurred or not in that plant compared to the mother plant. So for screening of desirable traits, it is done. Desired traits means what type of characters you want, what type of specific characters you want, and then after observing the desirable traits, we select the somaclonal variants, and then we will, for certain agronomical trials are going on then for that particular plant. Okay, we can also go for gel electrophoresis that we normally use to separate the DNA based on their molecular weight. Okay, based on their size. So this is simplified form of the without in vitro selection. Okay. Now, what is the limitation of without in vitro selection is that we are not, it is a very enormous technique because we are not inducing any selective agents. We are not trying to find out that whether the plants are getting resistant to that selective agents or not, what type of variation we want. Okay, suppose there is excessive salt in that particular field, so we have to regenerate such plants which can tolerate the salt. So by in vitro selection, with in vitro selection, we will see later on that we are adding the salt as a inhibitor, okay, at a certain concentration obviously, to check out that which plants are getting resistant towards that salt. Then that plants are further grown, they are analyzed, and analyzed for their molecular DNA, like for example they are molecularly analyzed, like we do agarose electrophoresis, we do native phase to check that whether they are becoming resistant or not, they are different from the mother plant or not. So here that thing is not occurring. Okay, so there is no direct, the limitations of without in vitro selection is there is no direct and specific approach for isolation of soma clones without in vitro selection. So here simply we are doing culturing, and and finally we are at the last we are checking that whether variation has occurred or not. No such specific approach is being done here. Consequently, the appearance of a desired trait is purely by chance. That means we are trying to get such beneficial trait by chance. We are not applying any selective agents to check that whether that plant is becoming resistant or can withstand in that conditions or not. Okay, like the tomatoes which are nowadays grown on hot water springs in Norway. It is a purely it is a fully cold country that we know, but they are trying to grow tomatoes now. Now how they can grow that tomatoes? If you want to plant the tomato grown in here, that plant in the Norway, it will not grow. Now, so why that tomato plant grow there? It's because by tissue culture technique they have tried to make that tomato plant resistant to that cold condition. They have used certain conditions in the tissue culture for the tomato plant to check out which explants are growing in that extreme cold conditions. And so with the help of this they grow such type of plants that can withstand in that conditions in that particular country. So that is the with in vitro selection. But in without in vitro selection we are not using any selective agents. We are It is not a specific approach. Okay, it is not a specific approach or direct approach for isolation of soma clones. It is appearing by chance. The desirable trait may be desirable or non desirable also. It's purely by chance. Further, this procedure is time consuming, obviously, because to find out such trait, it is very, very time consuming and it requires screening many plants. We have to screen them. Many, many plants we have to do screening, the molecular analysis we have to do, cytological study we have to do. Now, with in vitro selection, isolation of soma clones with in vitro selection method basically involves handling of the plant cells in culture. You take the protoplast, you can use callus culture like microorganisms, and then we have to use certain selection agents. Okay, the cell lines are screened from the plant cultures for their ability to survive in the presence of a toxic inhibitory substance in the medium or under the condition of environmental stress. So here the cell lines, the plants that we are getting deriving from them, cell lines has been given if you are culturing the protoplast cell on that extreme conditions. Okay, so they are screened further. Screened is done, as I have said, we have to check their if there is some minor structural changes in their chromosomes or not by cytological study, karyotype analysis, or by screening in the by electrophoresis to check their DNA, or for the proteins or enzymes if some structural change occur or not. And they are screened after they are, if we can observe their ability to survive in the presence of a toxic or inhibitory substance in the presence in the medium. So we are adding some toxic or chemical inhibitory substance, not at high concentration. We will increase the concentration and we'll see that what concentration up to that plants are withstanding. Or we can make some environmental stress condition in that area. Okay, so that how the plants are growing. you'll see that most of the plants will die, or some plants will able to grow from that explant. Then that type of plants are grown, and further they are screened, that is they are grown for gel electrophoresis, for the proteins and enzymes, compare with the mother plant. Okay, and compare the DNA and proteins with the mother plant, and then we will further go through the process like hardening in the greenhouse, field in the field, plant in the field. Okay, so a diagrammatic presentation of in vitro protocol for isolation of disease resistant plant with in vitro selection approach has been given. So here in this figure, it has been shown that how with in vitro selection, we are with in vitro selection we are actually making our disease resistant plants. Let us see. So you can see a plant. Okay, wait for a minute, we have to see the line what is given here. Okay, so first we use a plant, we take explant from the plant. See on the left side, not on the right side now. First see on the left side, okay. So first we use a plant, and from that plant, mother plant, we use a explant. We take the explant from it. Then from the explant we culture that explant, we grow the explant into callus. That callus is proliferated, and we maintain that callus. So up to this much we do. Okay, on the right side, you see a pathogenic organism is also being cultured. That means this part is related with the microbiology part. So in different lab we are culturing the pathogenic organism that commonly cause disease to that particular plant. That means we want to make this plant resistant to this pathogenic organism. We want to make this plant, the plants that we will derive from the explant of this plant, to becoming resistant to this pathogenic organism. So we take out a pathogenic organism that commonly cause disease to the plant. We culture them as we normally do in the microbiology laboratory. We purify the culture so that only that pathogenic organisms occur in that culture medium. Then we do filtration and isolate the toxin that cause these plants to be affected. We isolate the toxins first. After that, what we do, we also check out what is the lethal concentration of the toxin that affect the plants most. So we check out the toxicity of the toxin from that microorganism which can affect these plants, which can affect these callus or tissues of these plants. That is the lethal dose, that is the lethal concentration of that plant, of that micro of that plants, that how much they can withstand. Then what we do, this is the interesting thing. So up to this much you have understood that on the left hand side we have just cultured the plants into callus, and we are generating, proliferating the callus, not we are growing them into shoots. On the right hand side we see that the pathogenic organism that commonly cause disease to that particular plant is cultured, and we finally isolate the toxin from that pathogenic organism that mainly inhibit that growth of the plants. And then we check out for the toxicity determination. We actually observe the lethal concentration of that toxin that affect the plants. Then what we do, let us see here what is given. Small piece of callus, plural form is calli, CALLI, in the medium with toxin. Now we use a medium having that toxin derived from that microorganism, and on that medium containing toxin we are placing the callus, small piece of callus. Then what we do? Several selection cycles are being carried out. Several selection cycle is carried out to check out which callus are growing in that toxic medium, right? Growing into shoots wellly. In this way, we apply different concentrations of toxins in the culture medium. On the basis of that, we have to check out and we have to tabulate the data, statistical data, okay, to check out the particular plants, the callas which are growing into plants in presence of that, what level of concentration of toxin is given to the medium. So after several selection cycles, now it is not so much huge compared to the without in vitro selection, because here we are just observing, we are screening those explants which has been completely tolerating the can able to withstand in that inhibitory substance or toxic substance given in the medium. Only that plants are being screened, selected. Then isolation of the tolerant calli, that means which callas are getting tolerated can grow on that culture, on that toxic culture medium. Then we do regeneration, that means shooting, rooting is being done, growing into plantlet. Then again on the and then again during placing them on greenhouse condition, we again do screening procedures, like we check out the DNA content of the variants, we analyze them through gel electrophoresis for their proteins or enzymes, okay, for their proteins or enzymes, okay, for check out that they are getting, they wellly truly they have getting that disease resistant character or not, and that are referred to as R0 plants or SC1 plants as given here, the nomenclature that we have studied. Then either through self pollination, because we want to maintain that progeny, that disease resistant progeny, so we do self pollination, and the progeny clones from each plants are again tested for checking that whether they truly they are getting, they withstanding in that toxic conditions or not, okay. Then we regenerate the plants which is now considered as R1, or if you do the nomenclature with SC, we write SC2 plants. Again we do agronomical trials. In this way we maintain the culture lines which are getting that disease resistant property. So such a wellly selectively way we are regenerating that disease resistant plant. In the same way, through different environmental stress condition, as I have said before, in Norway we grow the tomato plants. In the same way, that type of plants are also being grown through somaclonal variation technique, through with in vitro selection, okay. So as given here, what I have said, the differentiated callus obtained from explant is exposed in the medium to inhibitors. So certain inhibitory substance are given, like certain toxins is given to the medium, or antibiotics or amino acid analogs. Then selection cycles are being carried out to isolate that resistant callus, that means which callus are growing in that medium, and those callas are then regenerated into plants which are further screened against that toxin or pathogen or other inhibitor. The plants which are resistant to the toxin are selected and grown again by self pollination or by vegetative propagation, and subsequent generations are further analyzed for disease resistant plants against the specific pathogenic organism. In this way, disease resistant plant via somaclonal variation can be achieved. In the same way, for certain environmental stress condition, we can also grow the plants. Let us see the list of some disease resistant crop which are grown through this somaclonal variation. Now it has been little bit blurred. Not blurred, pixelated. Okay, I will give you this image. I am writing this. I am not writing. Sorry, I am saying this. Okay, rice plant. Suppose rice plant, the rice crop has been obtained through somaclonal variation for growing against this pathogenic organism whose name is Helminthosporium. Helminthosporium oryzae. I'm writing the name here. Sorry again. Helminthosporium oryzae. So this is a pathogenic microorganism which damage the rice plants. So we, through somaclonal variation, rice plants are grown in this grown having this toxic agent obtained from Helminthosporium oryzae, and we grown such type of rice plants. Then for the maize, I'm writing just a name. For the maize plants can also be obtained through somaclonal variation, which are resistant against Helminthosporium maydis. Helminthosporium maydis. Okay. Then barley. For the barley plant through somaclonal variation, we obtained such barley plant which are resistant against Rhynchosporium sicalis. Rhynchosporium sicalis. Okay, this is you, this is I. Rhynchosporium, RIUM, sicalis. This is a pathogenic microorganism that damage the barley plant. So through somaclonal variation, we obtain the such barley plants which are resistant against Rhynchosporium sicalis. Then other plants are also are there like potato, which are getting through somaclonal variation we make them potato plant resistant against potato virus X and Y. Then Alternaria solani, Phytophthora infestans, this type of plants are being grown nowadays. Okay, so beside this disease resistant plant which has been described here, plants with herbicide resistance and antibiotic resistance has also been developed. Now, herbicide, you know, there are certain chemicals which will kill away your weeds. Okay, the plants which are affecting the growth of the crops. So, for that, we apply herbicide. Okay, now that herbicide will not only affect that weeds but it will also affect the crop plants. So, for that reason, we through somaclonal variation, herbicide resistant plants are also being grown nowadays. Okay, an antibiotic approach has also been shown here. Now, this is a selected list of disease resistant plant. Okay, so this is a selected list of disease resistant crop plants which are obtained from somaclonal variations with in vitro selection along with the pathogenic organism, and selective agents are also been shown. Like, for example, crop plants, rice, the pathogenic organism which has been used to make the rice plant resistant to them is Xanthomonas oryzae, and the selection agent is used as bacterial cells. So directly we culture the bacterial cells, we obtain and we the bacterial cells are grown in a medium. The rice plants through tissue culture we grow the callus, then we use the small piece of callus to grown on that medium having the bacterial cells, and we have to show them that which callus are growing in that conditions. In the same way, wheat, Helminthosporium sativum, crude toxin has been taken from that Helminthosporium sativum, and that crude toxin is allowed to culture on the culture medium, and on that culture medium the wheat callus are grown, and see screen further that which callus are growing in that medium having that crude toxin. So this is a selected list that we can normally see. I'm not talking all the names here. Okay. Now, what are the advantages of with in vitro selection? The advantages of with in vitro selection is that we are using selection method. It is directly we are selecting. It is of specific selection we are applying for our desirable trait. There, in without in vitro selection, it was fully the trait that we obtain is by chance. But here we are directly using selection agent for obtaining our desirable plant. Okay, and this procedure is less time consuming compared to the without in vitro selection way, and this is the main advantage actually with in vitro selection, that selection procedure time is less less consumed, and second is we are using selective agents for getting specific plants having that desirable traits. Now, what are the factors that affecting the production of somaclonal variation? What are the factors? Let us see. First is genotype and explant source. What type of genotype we are using? That depends on your somaclonal variation. The nature of genotypes and of the plants influences the frequency of generations and frequency of production of somaclones. That means what type of genotype, what type of explants we are using, depending on that, the genotype contained within that explant will affect your variation in somaclones. Explants taken from any part of the plant like leaf, root, internodes, ovaries, the source of explant is very critical for somaclonal variation. Like uh, on which what source we are using, the meristematic source or non-meristematic source. As I have said that meristematic explants which are derived from shoot apex or from axillary bud, they have less degree of genetic variability compared to the non-meristematic explants, because cells of meristematic explants divide by normal mitosis and they are maintained at diploid level, while the cells of non-meristematic explants, they which are the derivatives of meristematic explants as well, they do not divide by normal mitosis and they undergo DNA duplication or endo reduplication, which leads to chromosomes having which leads to the formation of chromosomes having 4 chromatids, 8 chromatids, just like protein type of condition, protein chromosome like condition, it will occur. Okay, it has been said here. Okay, I have not said. It said that the source of the explant is very critical for somaclonal variation. What type of source we are taking, that is very critical for somaclonal variation, like the potato plants which are regenerated from the callus, the potato plants which has been regenerated from the callus of rachis and petiole, they show much higher degree of variability compared to the those which are regenerated from the callus of the leaves. Rachis are actually the stem part of the plant, here the potato plant stem, that bears from the stalks at your start intervals. Okay, and petiole, as you know, the stalk that joins the leaf to the stem, the stalk which joins leaf to the stem, that part shows greater degree of variability in potato plant. If you want to take explant from the potato plant, and the explant is taken from the rachis of the potato plant, which is the stem of the potato plant that bears your that bears the stalks at the start intervals, or if you take the petiole of the plant, petioles are the stalk that joins leaf to the stem, they show greater degree of genetic variability. So depending on what type of source we are using, then duration of the cell culture also affect the variation among the plants. Like in general, for many plant cultures, somaclonal variations are higher with increased duration of culture. So if you do multiple subcultures for lengthy period of time, it can lead to somaclonal variation. Like for example, it was reported that genetic variability increased in tobacco protoplast from 1.5 to 6% by doubling the duration of the culture. So if you extend your culture periods for length for long period of time, it can also lead to variation. Now that variation is without in vitro selection. It may have desirable effects or it may not have. Growth hormone also affect your somaclonal, can also affect the variation in the plants. The plant growth regulators in the medium will influence the karyotypic alterations in culture cells and therefore development of somaclones, like your 2,4-dichlorophenoxy acetic acid, which is 2,4-D, and naphthalene acetic acid, which is NAA, which are frequently used to achieve somaclonal variations. It has been reported that growth hormone or the plant growth regulators which are used in the culture medium also influences, means affect the karyotype alterations, means the chromosome alterations in the culture cells and development of the somaclones, like your 2,4-D, which is 2,4-dichlorophenoxy acetic acid, NAA, naphthalene acetic acid, full name is alpha naphthalene acetic acid. They are frequently used to achieve this type of variations. Besides factors discussed here, selection of somaclones in vitro selection are dependent on the following parameters. What type of propagule you are using, what type of you are using, the cells or protoplast or the callus, from what part you are wanting them to regenerate into plant, depending on that. Okay, it also affects your variation in the chromosomes. Selection agents, obviously, what type of selection agents you are adding to the medium, okay, to grow that plant to withstand in that selection agent. Select techniques used for selection, stability of the resistant substance, in vivo testing, and ability for regeneration of the plants. Now the applications of your somaclonal variation, let us see. Okay, first is production of agronomically useful plants. So through somaclonal variation, we have make such type of plants which are agronomically very useful. Like as a result of somaclonal variation, several crops, different varieties have been developed. Like for example, pure thornless blackberries has been produced. Okay, several crops has been grown having this type of desirable traits, like crops induced include rice, wheat, maize, sugarcane, potato, carrot. Let us see, like for example, see the sugarcane. Okay, sugarcane. Through somaclonal variation, we have induced some mutations, certain changes in the structure on the structure of the their chromosomes that affects their trait. What type of change has occurred? High sugar yield, increased stalk in length, that is desirable. Increase in stalk length means more sugar content will occur, diameter, weight, and density. So that's a good thing. Through this variation, we can make this type of sugarcane plant, which of increased commercial importance. Then let's see the soybean. In soybean, we have induced them to make the variable height, maturity, seed protein, oil content, because soybean oil is extensively used nowadays throughout the world. So increase in oil content has also been induced in soybean through somaclonal variation. Potato plant, you know potato, we eat potato throughout the world. Higher yield, such type of mutations, some variations has been done in potato to increase their yield, growth habit, maturity, and morphology. Growth habit is normally is based on the seasonal type of change. Okay, so we are growing the potato plant normally grown in certain seasons. We store them in cold storage, but nowadays through somaclonal variation we can make this potato plant grow throughout the year. Okay, their maturity is getting much shorter period of within shorter period of time they become mature by variation in their chromosomes. Tomato, dwarf habit, early flowering, orange fruit color. So this type of carrot, highly carotene content, that makes them more red, more tasty, and more vitamins it will carry. So this type of things are being induced. This type of traits we get, beneficial traits we get through somaclonal variations, as you can see. I'm just um, not saying all the things. Okay, so this type of beneficial characteristics we can observe if we can able to achieve such type of selective traits through somaclonal variation. Second thing is resistant to disease. One example I have already been have said in the flowchart in the previous pages. Somaclonal variation has been largely contributed towards development of disease resistant in many crops like rice, wheat, maize, sugarcane, tobacco, apple, and tomato. These type of plants are employed for disease resistance normally nowadays. Selected crops somaclonal variants with increased disease resistance develop without in vitro selection and with in vitro selection are respectively given in table 46.1 and 46.2. 46.1 shows the morphological character. This is with in vitro selection. These all plants has been done having this beneficial characteristic with in vitro selection. For without in vitro selection is given in 46.2, that is in the next page. Yeah, this one. The 46.4, sorry for that. Mhm. This is 46.2. Okay, this is of without in vitro selection. Sorry, this is with in vitro selection. With in vitro selection, this type of disease resistant plant has been developed. And for without in vitro selection, okay. Resistance to abiotic stress. It has been observed possible to develop biochemical mutants having abiotic stress resistance. Like other than disease resistance, we can also put the plants in such a stress condition, abiotic stress condition, that they can able to withstand, and we can grow them and selectively marketized as those plants. Like freezing tolerance, example wheat. Wheat are nowadays cultivated in cold countries as well nowadays, but that wheat is, we are getting them through tissue culture through somaclonal variation by testing that wheat callus or the protoplast that we obtain from the culture conditions in wheat in freeze conditions. Okay, salt tolerance plant like rice, maize, tobacco can be produced. Aluminum tolerance like carrot, sorghum, and tomato. So these type of abiotic stress plants can be produced through somaclonal variation. Then resistance to herbicide, I have already said. Certain somaclonal variation with herbicide resistance have also been developed. Some selected examples has been given here. Okay, like certain your tobacco plants which are resistant to glyphosate, which are commonly used herbicide used in western countries. This glyphosate, sulfonyl urea, and picloram. These are the certain your herbicides which are normally employed in the field and to kill away the weeds. Okay, but and it can also affect the crops as well, because the same toxin is applied to the crop plant also, spread to the top crop plant. So to withstand on that herbicide condition, we can grow certain plants having that resistant property through somaclonal variation. Carrot resistant to glyphosate, lotus which are resistant to 2,4-dichlorophenoxyacetic acid. It is not a toxic chemical, it is a phytohormone which are being grown. Improve seed quality. We can improve the seed quality through somaclonal variation like the plant Larythus sativa. Larythus sativa is commonly known as Khesari plant. Okay, labri. In Bengali it says Khesari. Normally it says Kesari or Khesari. These type of seeds are commonly used in old days. It's also used nowadays also in certain rural areas. So certain variety of Larythus sativa seeds has been developed through somaclonal variation having low content of neurotoxin. Now it has been reported that Khesari plants, which the seeds that we obtain from Khesari plants, they can be used as your legume, okay, as pulses, as dal, okay. In Bengali, we in rural areas they mostly eat khesari dal in old days, but it reported has been shown that khesari seeds have high amount of neurotoxin, which can cause different disease, okay. Mostly in central part of India, this type of seeds are being eaten still now also. But so for that reason, scientists have tried to improve the variety of these Larythus sativa seeds by lowering the neurotoxin content in them. Okay, and thus it's a good thing. We can use them now in our normal consumption of as a food in all over India. But all the things have some limitations, so also here somaclonal variation have some limitations. First, most of the somaclonal variation may not be useful. This is the first thing. The somaclonal variants that we are getting, it may not be useful in case of without in vitro selection, right, or purely by chance. Variation occurs in an unpredictable and uncontrolled manner. So the variation that we get, suppose disease resistant plant, they may be unpredictable or uncontrolled manner. That's the second problem. So much of investment of money for making somaclonal variation, it may not be, it is unpredictable and uncontrolled manner. It may affect the normal plants as well. Third, many times genetic traits obtained by somaclonal variation are not stable and eventually they are not heritable. So the genetic traits that we get, the characters that we get from somaclonal variation may not be stable or heritable. That's a big problem. If it is not heritable, then we have to invest money, invest our all the labor stuffs for obtaining that type of variations, which we may not we get next time. Somaclonal variation are cultivar dependent, which is frequently a time consuming process. Yes, we need labor. It is a cultivar dependent process. Somaclones can be produced in only those species which regenerate to complete plants. So it can only be obtained, somaclones can only be obtained which can regenerate to plants. Like for instance, complete plants means what? Plants grown fully and they are capable of reproducing. Like for example, you can grow apple plants in your garden also, but have you noticed that that type of apple plant cannot grow into fruit? That is not a complete plant. Okay, complete plant means which can able to produce its progeny also, as well as it can grow to a certain height, the normal height that normally they grow. So somaclones can only be produced in only those species which regenerate fully to complete plant, and many cell lines may not exhibit the regeneration potential. Many cells they cannot capable to grow them into complete plant. So this was about the somaclonal variation. Now one extra term, gametoclonal variation. Well, it was almost it is 1 hour. So what is gametoclonal variation? The variation observed while culturing the gamete cells. If you culture the gamete cells, the pollens, the ovaries, okay, and the variation you get from those gamete cell during culturing can be regarded as gametoclonal variation. As simple. In contrast to the somaclonal variation detected in cultures of somatic tissues. That's why we write somaclonal variation, because we obtain the somatic tissues. But gametoclonal variation is when we are culturing the gametic cells, and the variation we get from that gametic cells can be regarded as gametoclonal variation. Now, as somatic cells divide by normal mitosis, the genetic material is equally distributed to diploid level, and it is equally distributed to the daughter cells. Because in mitosis, the genetic material equally divided into daughter cells, but in gametes, being producer of products of meiosis, they possess only half of the parent cell genetic material. So little bit of variations you can see. Gamete cells, they are the derivatives from my meiosis products. So that's why they only possess half of the parent cell genetic material. The somaclonal variations differ from the gametoclonal variations differ from the somaclonal variation by three distinct features. First feature is what? Mutants which are obtained from gametoclonal variation give rise to haploid plant, since a single set of chromosomes are present. Now we have seen the haploid plant production, the anther culture and pollen culture and the ovary culture, the androgenesis, the gynogenesis concept, where we can generate a haploid plant. So here the mutants that we obtain from the gametoclonal variation, they are haploid plants. They have half sets of chromosomes, single sets of chromosome present. Second difference is meiotic crossing over in recombination process observed in gametoclonal variation. So meiotic crossing over is the recombination process observed in gametoclonal variation, and the third is the gametoclones can be stabilized by doubling the chromosome number. So we have to increase the number of chromosomes. Okay, we have to double the chromosomes, then only we can stabilize the gametoclones, because if we induce some variations, then they cannot able to withstand, because they have only single set of chromosome, the other set of chromosome is missing. Not missing, actually naturally it is missing, because the gametes obtained from meiosis products. So for that we have to doubleize the gametic chromosome number, then only it can become stable. We can get the trait also, and we can grow the plants also. Okay, so this was all about the somaclonal variation, and I hope you have understood this part. If you have any queries, any questions regarding this, you can say in the WhatsApp group. I will try to explain the answers. Okay.